All right, whenever you're ready.
I'm ready. All right, we're going to do the immune system today. We're going to try to get through as much of it as we can in one day. So again, I'm going to be moving fast. It's a review. I don't know if you guys have those outlines that were a handout. I'm going to follow that. It's just a condensed form of our notes. There might be a couple up there on the card. So, all right, so we know there are two major big sides of the immune system. One is the innate or non-specific, and it is what it says it is. It's non-specific. It doesn't care if the measles is in. It doesn't care what it is. It's going to take care of anything non-specific. The other side is the specific or the adaptive. That is the side that creates the antibodies to the measles or gets very specific when the antibodies go after the antigen. So we're going to talk about the innate side first. We're going to move quickly through that. It's not very difficult. So we have our basic anatomical barriers. Skin. Think anything with the skin. Sweat, the hair on our skin, anything that can create a barrier to the bacteria. Chemical on the skin, we have sweat, and we have something called lysozymes. We find that in a lot of different places, mucous membranes, sweat, tears, and lysozymes are bactericidal. So they help take care of the bacteria. Again, it's not specific. They don't care what kind of bacteria it is. And then biological, colonization of normal flora. We never talk about our normal flora, and it's very, very important. Our normal flora helps battle any of the pathogens by competing for food. So our normal flora is really important. We also have components that are inside. If the outside doesn't take care of the organism and they get inside, we have some components inside. Some of them are complement. Now I know when you were in micro we talked about complement until you were like nauseated with it, but complement is a protein system. It's a number of small proteins. They're usually listed C1, C2, all the way through C9. Each one of those C's has something to do. They are cleaved when the complement system is activated, and then each one of those C1a and C1b have something to do. They're usually synthesized by the liver. That's something that we didn't talk about in class, and I think that's a great question for the board. So complement synthesized by the liver. They're circulating all the time, and they become activated in different ways. We're going to talk about that a little bit later. Some of the things that complement do. Opsonization. I don't know how many times we said that during micro 2. Opsonization is putting cheese on the broccoli. The broccoli is the antigen. You want to put something on the antigen to make it more palatable to the phagocytes. Now, in real life, opsonins are either complement or antibodies. And they coat the antigen, and it makes them more palatable to the phagocytes. So complement are opsonins. Chemotaxis, they also attract macrophages, neutrophils, anything that we need for that area to take care of that antigen. They help with cell lysis, they rupture membranes of foreign cells, and they also help with clumping of antigen-bearing agents. So they'll help clump antigens and get rid of them. They also trigger inflammation, fever, which of course when we have an infection is a good thing. We want inflammation, we want fever, and they, by doing all this, they help destroy foreign antigens or bacteria or viruses. The coagulation system, also found inside. The coagulation system increases vascular permeability. Again, why do we want that? We want that because then we can get things where they need to go. If the vascular system remains closed, we can't get cells to where they need to go. So it increases vascular permeability. The platelets that go to the site of the injury release this beta-lysine that is a little bactericidal. They also help recruit phagocytic cells when the platelets all kind of clump together. Lactoferrin and transferrin, these are chemicals that compete with the pathogen for iron. The pathogens need iron. If they can't get iron, they're going to die. So again, that's part of our good normal flora thing. Lysozyme, we talked about that. It helps break down cells. And cytokines. Cytokines are signalers. If you can remember signalers, the ones we talk about mostly in our immune system are the interleukins. Interleukins are a type of cytokine. Generally, we talk about cytokines, interleukins. Some of the other ones are interferons, growth factors, and chemokines. In our notes, we just talk about the interleukins. All right, cellular components. Again, cellular components on the inside, they all have a job. And again, we're still talking about nonspecific. Neutrophils. Neutrophils are a white cell. Our white cells, of course, are our immune system. Anybody remember what is the job of neutrophils? Please, somebody tell me.
Break down.
To bacteria, to kill bacteria. If you don't go away with anything else, that is just a favored question on every healthcare board. Neutrophils kill bacteria by phagocytosis. That is their job. Monocytes are in the blood. They're there for a couple days, and then they get into the connective tissue and become macrophages. So monocytes also intra and intercellular killing by phagocytosis. And then the macrophages have a job in the adaptive immune response. Natural killer cells, they are a type of lymphocyte. They're not a T cell, they're not a B cell. They are considered part of the innate immune system because they are not MHC restricted. They do not have to work with MHC in order for them to kill antigens. You remember, we'll get to it, but you remember all that MHC stuff. They are not MHC restricted, so they're part of the innate immune system. And eosinophils, on that outline, I don't have basophils there, but eosinophils work with parasites, and they also have a job, not a job, but they have a role in our allergies. And then basophils, I neglected to put basophils, don't forget them. They have a role in allergies also, especially in the anaphylactic shocks and the type 1 hypersensitivities. All right, the adaptive immune system. It reacts with a specific antigen. That is one of the hallmarks of the adaptive immune system. It is specific. If we come in contact with the measles, it's going to create antibodies to the measles specifically. We have a cell-mediated side and then a humoral side. Cell-mediated, it's where the cells are the effector cells. The cells take care of the antigen. The humoral side, they rely on antibodies, and it's humoral because the antibodies are inside of our humors or our liquids in our lymph, in our blood, and they float around in there. So the cell-mediated side, it involves phagocytes. That also has to do with natural killer cells, cytotoxic T lymphs, and then our humoral side, B lymphs, which form plasma cells, which secrete antibodies. So if they ask you what is involved in humoral immunity, what are you going to tell them?
Antibodies.
Antibodies, definitely antibodies. All right, the hallmarks of the adaptive immune system. There has to be recognition of self and non-self. All right, we have to be able to tell the difference between self and non-self. If we can't, and our immune system attacks our self cells, what is that? Autoimmunity, yeah. So and it happens, but in a perfect immune system, we don't want it to happen. Memory in the adaptive side. Memory is a huge thing. It remembers when we've come in contact with an antigen or the measles, so that if I come in contact with it again, the reaction is a lot stronger, it's a lot faster, and usually you don't become ill or you become just very mildly ill. Anybody remember what the memory cells trigger? Anamnestic response. I don't even have to ask the question. So the memory cells, when I come in contact with it again, trigger the anamnestic response. And that's the second time I come in contact to an antigen. Specificity is a hallmark to the adaptive immune system. Again, it's very specific. And diversity. It's very diverse. We have we've got billions of different antibodies that we have floating around in us, and it's very, very diverse. All right, so the immune tissues that are involved in this, we have primary lymphoid organs. These are the bone marrow, the thymus. In the fetus, it's the fetal liver. If you just go with the bone marrow and thymus, I think you'll be okay. This is where the cells become immunocompetent. Immunocompetent means that they are able to create a normal immune response. So the cells are born in the bone marrow. The T cells take a trip through the thymus. The secondary lymph tissue is where they go to work. That's where they're going to hang out. After they become immunocompetent, they're released from their schools and they go to the spleen, the lymph nodes, the tonsils, and all of that mucosa-associated lymphoid tissue. We have it in the bronchus, we have it in the skin, we have it in the gut. Anywhere there's lymphoid tissue, the T cells and the B cells go there, they hang out. These are perfect spots for them to come in contact with antigens. So they are born and they go to school in the primary lymphoid organs, they go to work in the secondary lymphoid organs. All right, again, I'm gonna skip this part about the white cells, the monos, the grands. We just talked about that stuff. One thing, eosinophils do produce histamines. Eosinophils have a role in our allergic reactions, and if you remember the commercials on TV, they're always talking about antihistamines. So they're trying to get rid of those histamines. Basophils will release histamine and heparin. Histamine seems to be the big one that they talk about, but kind of try to remember heparin in the back of your mind also. All right, we're going to talk about lymphocytes. Natural killer cells, we talked about them. They're part of cell-mediated immunity, and again, they're considered part of the innate immune response because they're not MHC restricted. That's another favorite question in every health board that there is. B cells are involved in humoral immunity. They mature in the bone marrow. They go to school. They're born in the bone marrow. They go to school in the bone marrow. And then they go out and hang out in the secondary lymph tissue. They become plasma cells, and then the plasma cells create or secrete antibodies. That's what they do. T cells are involved in what we call cell-mediated immunity. They mature in the thymus. They're born in the bone marrow. They mature in the thymus. That's why they're called T cells, mature in the thymus. And then they go out and hang out in the secondary lymphoid organs. Their main job, they secrete cytokines. Once they've come in contact with an antigen, they secrete cytokines, and they send those cytokines to the B cell to say, make this type of antibody. And they also have two co-receptors. T cells not only have a T cell receptor, they also have co-receptors, a Cd8 and a Cd4. The Cd4 are the T helper cells, and the Cd8 are the cytotoxic T limbs. These co-receptors come into play when antigens are recognized. All right, some vocabulary. An immunogen. It's anything that triggers an immune response. All right, anything that's going to trigger a response. Now, the difference between an immunogen and an antigen can bind with that response, but it may not be able to trigger a response. So we kind of use antigen all-encompassing. It's kind of, we should talk about immunogens, but we kind of change the wording around. So some of the antigens are too small. They have to be coupled with a carrier in order for them to elicit an immune response. But antigens can always couple with that immune response. A hapten is an incomplete antigen that needs a carrier. It's too small or not complex enough and has to be bound with something else in order for it to create a response. An adjuvant, it's something that they add to immunizations, and it enhances immunogenicity. An epitope. Epitopes are kind of important. The vocabulary is important. It's an antigenic determinant, and it is the part of the antigen that's recognized by the immune system. So when the antibody comes up and says, okay, I recognize you, you bad antigen, it's not going to recognize the whole thing, but it's going to bind to a part on the antigen, and that is the epitope. I kind of think of it as a little ball on the outside of the bacteria or whatever. It's going to bind to that part. So that's the part of the antigen that's recognized by the immune system. And then an allergen is any substance that can cause an allergy. All right, factors that influence immunogenicity. I'm going to go over this real quick. Foreign. The more foreign, the better. That's the way our immune system is set up. Size. In this case, size is better. Bigger is better. Usually, the larger the antigen, the more immunogenic it's going to be. Chemical compositions, again, the more complex it is, the better it's going to be. Physical form, remember we have to bring it into the macrophage and it has to be degraded, so particulate antigens are better than soluble antigens. And then degradability, it has to be able to be degraded and presented as a peptide. If it can't do that, it's not going to be a great antigen. Genetics, genetics is very important in the immune system. Age, the very young and the very old are more susceptible to things. Method of administration, we're talking about vaccination. The dosage is important, too much or too little, and you're not going to get the correct response. And the route, subcutaneous is usually better. So now they have the nasal sprays for the flu. They used to have the oral for the polio. I don't think they give that in the United States anymore. And then adjuvants. If the adjuvants are added, they're supposed to create a nice, stronger response. Chemical nature, proteins, they're very complex. They are the best immunogens. They can be pure proteins, glycoproteins, lipoproteins are okay, but pure proteins are better. Usually the proteins are very good. Nucleic acids aren't very good. Remember proteins as being very good. All right, antibodies. What are antibodies? AKA immunoglobulins. So don't get confused if they ask you about immunoglobulins or antibodies. Use them interchangeably. The abbreviation is a big I with a little g and then another letter after it for the class. They are gamma globulin proteins. They are a type of protein in our blood. They're found in our bodily fluids, and they identify and neutralize foreign antigens. That is their job. So they are gamma globulins. They're called immunoglobulins or antibodies. They're all the same. How they're acquired, I'm going to talk about that later. I'm going to come back to the natural, artificial stuff. The structure of the antibody, it is a Y configuration. Okay, Y configuration. This is the FAB portion, and this is the antigen binding portion. This is what holds on to the antigens. My feet is the FC portion, or the constant portion. That is what binds to receptors on cells. Okay, so antigen binding and constant portion, that's what binds to receptors. Let's see. Okay, that's real quick. The heavy chains of the antibodies are what determines what class the antibody is. That's something, it's not spelled out in our notes, but it's something I would ask if I was writing for the boards. So we know they're comprised of heavy chains and light chains. The heavy chains are the ones that determine what class it is. All right, classes of antibodies, IgG, and you know antibodies, GAMED, gamed. Those are the types of antibodies in mammals, GAMED. So IgG is the most abundant. It's the smallest, so in other words, that makes it able to be transplacental. These are the antibodies that the mother gives the baby in utero. They're transplacental. IgG is also the first antibody to be made after a first encounter with an antigen. All right, the very first one. No, strike that, nope, that's wrong. M is the first one. All right, yes, strike that. All right, secretory IgA. Secretory IgA found in mucus, mother's milk, tears, the GI tract. It kind of floats around a little bit, and they put a little S in front of the IgA. And then we also have serum IgA that floats around in our serum. But the secretory is found in tears, milk, the mucoid material in the GI. IgM, here we go. The first one made following an antigenic stimulus. So when I am first confronted with the measles, M is first and G is second. Remember my good. Campbell soup is good. So M first, G second. Upon a second stimulation, or an anamnestic response, then what antibody then is the first? G is first. It's the good antibody. And this G is very good. It's usually lasts longer. It's a higher in titer. So the first response, it's M and then G. The second time you come in contact with it, it's G. IgD. What I would remember about this, it's primarily found on the B cell surface, and it's found in the B cell receptor. So what is that B cell receptor then? It's an antibody. Okay, so IgD found in the B cell receptor. And then IgE, and I have involved in allergic reaction, also in parasitic infections. Those two things, an allergic reaction and a parasitic infection, will create a lot of IgE. All right. Some of the terms for antibodies. Again, if you can't remember exactly what it means, know that this has to do with antibodies. And we use a lot of these in conjunction with testing. So an agglutinin is an antibody that causes an antigen to agglutinate or clump. Again, it's great to use for testing. We can just do the testing. If it clumps, then we know it's there. A precipitin is an antibody that causes an antigen to precipitate out of solution. Lysins, that's easy. It's an antibody that lyses an antigen. An antitoxin, again, anything with anti in front of it means an antibody to whatever it's in front of. So an antitoxin is an antibody to a toxin, and it's a soluble antibody that neutralizes toxins, especially tetanus, diphtheria, and botulism. Those are issues that cause us issues because of the toxin itself, not because of the bacteria. So we have to neutralize that toxin. Affinity talks about the strength of one single antigen determinant and a single antibody combining with it. Avidity is the overall strength of multiple sites. How strong are these multiple sites? Specificity, again, how specific is it? It's the ability of an antibody to discriminate between two different epitopes. It's specific. Our immune system is very specific, but there is cross-reactivity, where the ability of an individual antibody to react with more than one antigen. And a great example of this is cowpox and smallpox. The smallpox immunization is made from the cowpox virus, but because of this cross-reactivity, if we're exposed to smallpox, the antibodies to the cowpox will take care of it. There's a cross-reactivity there. All right, B and C cell development. In our bone marrow, there are cells that will create any of the hematology cells, the blood cells that we need. Hemocytoblast is one word. I would remember stem cells. These are stem cells. They can create any kind of cell in our blood that we want. So the T lymphocytes, let's talk about T lymphocytes. They are born in the bone marrow, they take a trip to the thymus, and then they go to the spleen and the lymph nodes. This is considered cell-mediated immunity. If they ask you what type of immunity is concerned with T cells, it's cell-mediated immunity because the cells are the effector things. In humoral, the antibodies are the effector molecules. In the cell-mediated, it's the cell. They also, though, have a job in humoral immunity, and that is to help the B cells know what type of antibodies to make by sending cytokines to them. So they do have a job in the humoral immunity, but their main role is cell-mediated. All right, cytotoxic T lymphs, CTLs, cytotoxic T cells, or Cd8 cells. And the Cd8 is named for a co-receptor on the cell that when we have an antigen, when we come in contact with an antigen, that Cd8 grabs the MHC to kind of hang on to it. So Cd8, sometimes they're called cytotoxic T lymphs, sometimes they're called Cd8 cells, used interchangeably. I would think they would use cytotoxic T lymphs on your test, but I'm not 100% sure. These cells appear very, very early in the immune response, and they release granules to attack viruses and tumor cells. early in the response. We want to get rid of those cells as soon as possible. I do not want cancer cells hanging around. I do not want virally infected cells hanging around. The cytotoxic T lymphs come in and destroy the whole cell. It's cell mediated. They destroy it by something, one of the things is called perforin. It's not on our notes, but it perforates the target cell, so perforin. All right, our T helper cells, or our CD4 cells, they interact with antigen before the B cells interact with antigen. They actually tell the B cells what type of antibody to make. They help B cells synthesize antigen or antibodies by sending cytokines, specifically interleukins, to the B cells and say, make an antibody to the measles because we've got measles out there somewhere. They also help stimulate the activity of cytotoxic T lymphs. Now, mentioning under our T cells, we have suppressor cells. I think now they're called T-regs, or regulator cells. They regulate the immune system. Once the immune system is active, these are active also because at some point we have to dampen that immune system or it will get out of control. All right, B lymphocytes. They go to school, they're born in the bone marrow, then they go out to the spleen and the lymph nodes. They are involved in humoral immunity, which means they make antibodies. Again, the humors are the liquids of our bodies, and the antibodies, it's a great place for the antibodies to go so that they can move around and find any antigens that have invaded our bodies. The B cells, okay, upon stimulation, they form effector plasma cells. So B cells, once they get the cytokine, the interleukin from the helper cells and say, make an antibody to the measles, they form plasma cells, which secrete antibodies, but they also form memory cells. Don't forget about the memory cells. That happens with the T cells also. We're left with T memory and B memory cells. All right, so we need to have, for converting a B cell to a plasma cell, we have to have a macrophage, or some kind of antigen-presenting cell. We have to have T helper cells because they help the B cells. We have to have exogenous antigen. What is an example of exogenous antigen? Anybody remember? Bacteria. MHC class 2, because they deal with exogenous antigen. We have to have cytokines, more specifically interleukins, and we have to have the B cells that are going to make the plasma cells and the antibodies. All right, so when your class was in Micro 2, did we use the thing to remember the 4 and the 2 and the X and the P? Mm-hmm. All right, my suggestion is to go over that. My suggestion is somehow remember that. If you have scratch paper when you take the boards, write it on something, and it will give you answers to MHC, antigen-presenting cells, endogenous, exogenous, all that stuff.
1N8A.
Say that again?
1N8A.
Yeah, it's. I can do it after class or after this if you want. I'll write it on the board. I don't want to take the time now, but I'll write it on the board, and it's just an easy way to remember that. If you write it all down, you will have answers to every one of those questions. So, all right. So let's talk about MHC. MHC are molecules on the antigen-presenting cells that are like a flag, and they hold the antigen and wave it like, here, I have an antigen. I need somebody to come along and make something to kill this. So MHC1 is found on every nucleated cell in the body, or I say any nucleated cell. And I use any because it goes along with those pen stroke things. So any nucleated cell in the body. They present endogenous antigen. Endogenous antigen is created within the cell, and it changes the machinery of the cell. The two main things that do that are cancer cells and viruses. They change the machinery of the cell. Bacteria don't do that. They come in and they're killed and they're gone. But those two things change the machinery, so they are endogenous antigen. They present the antigen to cytotoxic T lymphs. Cytotoxic T lymphs are the things that are going to deal with antigen or with viruses and cancer. Those are the things they're going to create that perforin, and they're going to kill the whole cell. And then activated cytotoxic T lymphs secrete toxins. And again, if you want to write perforin there, that's an example of one of them, and cause lysis or apoptosis of the whole cancer cell. Again, we don't want those viruses and those cancer cells left alive. We want them gone. All right, MHC class 2, only found on the professional antigen-presenting cells. And I use the P there in that pen stroke thing, the professional ones. They are B cells, macrophages, and dendritic cells. Dendritic cells is one of the more important ones. It's found a lot, so I don't know. Remember all of them. They're all fair game, but dendritic. These are the professional antigen-presenting cells. They present exogenous antigen, and an example of exogenous is bacteria. And they present to T helper cells. And then the T helper cells activate the B cells by producing cytokines, specifically interleukins, to tell the B cells to make memory cells and antibodies. Okay? All right, let's talk a little bit about the antigen-presenting cells, the professional ones. Again, dendritic B cells, macrophages. I don't know if there's any way you guys have come up with to remember that, DMB or something, but you have to remember them. Those are the professional ones. All right, processing. What does it mean when we process antigen? Basically, the antigens are processed into peptides. When they're brought into the antigen-presenting cells, they are processed, they're made into peptides. The MHC molecule grabs them, and then they're out on the surface of the cell, and they're saying, okay, I've got an antigen here. So they have to be degraded, they have to be processed and degraded, and then presentation. So the presentation is a peptide MHC complex, and it's transported to the membrane where it's displayed. Endogenous antigen, we've talked about this. It's generated in previously normal cells as a result of cell metabolism, viruses, cancer, and I'm going to name this, but don't get it confused with regular bacteria, intracellular bacteria. Do you remember the rickettsia and the chlamydia we talked about? Those are obligate intracellular bacteria. They change the machinery of the cell because they're using that cell's ATP, and they're reproducing in there. The big ones are the viruses and the cancer cells, though. Exogenous enters the body from outside. Think about a strep throat. When you breathe in, it enters from outside. It's a strep throat. It's just a bacteria. It comes into the cell. It's metabolized and it's killed. All right. It doesn't change the machinery of the cell. Bacteria don't have the capability to do that. So they just kind of they're killed. They come in from the outside. Think of a strep throat coming in. All right, all of the T cells have a T cell receptor. And again, both the cytotoxic T lymph and the T helper cells. The job of the T cell receptor is to recognize antigen. All right, so I have an MHC molecule presenting an antigen here. So my T cell comes up with the T cell receptor. They're going to hook on and dock, and the T cell receptor has to recognize that antigen. That is the job of the T cell receptor. It also has a little job in that it has to recognize self-information, which is in the MHC. Remember, we've got to determine self from non-self. So it has to recognize self too. The main job, though, is to recognize that antigen. It provides specificity for an individual T cells, and it recognizes a particular antigen. All right, so let's talk a little bit about a T helper cell, MHC class 2. All right, so we have a T helper cell. It has a T cell receptor, and it also has a CD4 co-receptor. All right, so T cell receptors will recognize antigen in